Hello everyone, welcome back to the channel. In the last video we had discussion in the second week of gestation, and in this video we'll be talking about the third week of gestation, what the different parts, gastrulation, notochord, clinical importance and the other stuff. My name is Dr. Zahir and I'll be talking about the third week of development. So what is biliminar disc? You know that it was discussed in the second week. In that situation there were two layers, that is the epiblast and the hypoblast, and the development of the trophoblast and the embryoblast. In gastrulation, the biliminar disc is transformed into the trilaminar disc, that is the ectoderm, mesoderm and the endoderm, by the primitive streak. The primitive streak is the cells that proliferate from the caudal end towards the cranial end, and these cells start from the caudal end towards the procaudal plate. So the procaudal plate is columnar cells over the hypoblast. The primitive plate towards the midline forms the primitive node, also known as the Helson's node, and towards the primitive pit or primitive groove. And over that part we have the amniotic cavity, and below we have the yolk sac. So this primitive streaks forms the anterior, posterior, left, right and the craniocaudal orientation of the embryo. So let's see what is the magic. Cells from the epiblast migrate towards the primitive node and primitive streak, go downwards and replace the hypoblast, forming the definitive endoderm. Then they spread out, forming the mesoderm, and then they completely replace the epiblast to ectoderm. This process is known as the invagination or ingression, and the new layer that is formed is the mesoderm layer. So let's see how it forms. The first cells that displaces the hypoblast and form the definitive endoderm, and then the next wave comes and they spread out and they form the mesoderm. Remaining cells of the epiblast, they form the ectoderm. So these are the three layers: endoderm, mesoderm and ectoderm. So the next part is the formation of the notochord. Before that, we'll see the fate. The ectoderm forms the skin, nervous system and sensory organs. Mesoderm forms the bones, muscles, blood vessels, kidneys and reproductive organs, and the endoderm forms the digestive, respiratory tract, liver and the pancreas. So how the notochord is formed? Let's see. Initially it is a cylindrical part. After it merges with the endoderm, there is a formation of a tunnel that is between the amniotic cavity and the yolk sac. The tunnel is important to give nutrition between the two parts. Then again it forms a solid structure after it gets displaced from the endoderm. In this diagram you can see first it is a cylindrical structure, then the connection is formed, and afterwards the proper notochord is formed. What is the function of the notochord? So it is helped in the induction of the formation of the neural tube, and the neural tube is formed. Afterwards they form the neural tube, we have CNS, and also it helps in the skeletal support. Here's another diagram showing the formation of the notochord, the sagittal section. You can see there are two diagrams. First of all, the diagram which is marked A is the area where the notochordal plate is formed, and soon the notochord will detach and form the diagram B. So in this diagram you can see the notochordal plate, how it forms, how it merges with the endoderm and then forms the definitive notochord. Let's see the development of trophoblast. We have primary villi, secondary villi and tertiary villi. Basically the primary villi, it is the cytotrophoblast which is surrounded by the syncytotrophoblast, and the secondary villi we have the mesodermal core, and in tertiary villi we have villus capillaris in the mesodermal core. In clinical we have caudal dysgenesis, also known as serenomelia, also known as the mermaid syndrome. So it was a quick review or revision of the third week of gestation. Do subscribe to the channel and give it a like. Thank you so much.